Hi everyone, welcome to NEET Biology channel. Which technique is best to develop hybrid seeds manually? A. Self-pollination. B. Cleistogamy. C. Artificial hybridization. D. Entomophily. Correct answer is C. Artificial hybridization. Explanation: artificial hybridization is the most effective technique for manual development of hybrid seeds because it involves the deliberate crossing of selected parents with desirable traits. This allows plant breeders to control the genetic makeup of the offspring. Self-pollination, A, does not introduce genetic variation and defeats the purpose of creating hybrids. Cleistogamy, B, where flowers remain closed and self-pollinate, also prevents hybridization. Entomophily, D, or insect pollination, is natural and not controlled by humans, making it unsuitable for precise hybrid seed development. A grower wants to prevent self-pollination in bisexual flowers. What should they do? A. Add sugar solution. B. Cut petals. C. Perform emasculation. D. Provide more light. Correct answer is C. Perform emasculation. Explanation: emasculation, the removal of male reproductive parts, anthers, is essential in bisexual flowers to prevent self-pollination and allow cross-pollination with a desired pollen source. Adding sugar solution, A, or providing more light, D, does not affect pollination directly. Cutting petals, B, might expose the flower but won't prevent the anthers from releasing pollen. Therefore, emasculation is the only method that effectively prevents the flower from pollinating itself. Which structure should be targeted to inhibit cross-pollination in a crop field? A. Anther. B. Stigma. C. Sepal. D. Ovule. Correct answer is B. Stigma. Explanation: the stigma is the receptive part of the female reproductive organ where pollen grains must land to initiate fertilization. To prevent cross-pollination, it is the stigma that must be blocked or protected. Anthers, A, release pollen and are more relevant in preventing self-pollination. Sepals, C, are non-reproductive and only protect the bud. Ovules, D, are located inside the ovary and are not directly accessible to external pollen, so targeting them wouldn't prevent cross-pollination. Which type of flower should be selected for crossbreeding to maintain genetic diversity? A. Cleistogamous. B. Chasmogamous. C. Wind-pollinated. D. Asexual. Correct answer is B. Chasmogamous. Explanation: Chasmogamous flowers open and expose their reproductive organs, allowing cross-pollination and thus promoting genetic diversity. Cleistogamous flowers, A, remain closed and promote self-pollination, reducing genetic variation. Wind-pollinated flowers, C, do allow cross-pollination but lack the precision and control needed for selective breeding. Asexual reproduction, D, does not involve pollination at all and results in genetically identical offspring. Bagging is ineffective in A. Insect-pollinated crops. B. Wind-pollinated crops. C. Cleistogamous flowers. D. Artificial hybridization. Correct answer is C. Cleistogamous flowers. Explanation: Cleistogamous flowers do not open at all, which means they automatically self-pollinate without exposure to external pollen. Therefore, bagging, used to prevent unwanted cross-pollination, has no role. Insect-pollinated A and vine-pollinated crops B benefit from bagging to prevent contamination by foreign pollen. In artificial hybridization D, bagging is an important step to control pollination events, making it very much necessary. Which of the following would not help ensure successful artificial pollination? A. Emasculation. B. Rebagging. C. Nectar removal. D. Selection of receptive stigma. Correct answer is C. Nectar removal. Explanation: nectar removal can actually hinder pollination, especially in species that rely on insect attraction, which is mediated through nectar. Emasculation A ensures self-pollination is avoided. Rebagging B is essential after pollination to prevent unwanted pollen from contaminating the stigma. Selecting a receptive stigma D ensures that the pollen has a suitable surface to germinate, making it vital for the success of artificial pollination. In a lab setup, pollen does not germinate on the stigma. What might be the cause? A. Overripe anther. B. Immature stigma. C. Too much light. D. Sugar concentration. Correct answer is B. Immature stigma. Explanation: pollen germination can only occur on a stigma that is physiologically mature and receptive. If the stigma is immature, it won't produce the enzymes and moisture necessary for pollen tube formation. An overripe anther A may affect pollen quality, but the primary issue in germination lies with stigma receptivity. Excess light, C, or sugar concentration, D, are secondary and do not directly control stigma-pollen interaction. A plant breeder wants to develop disease resistance in a crop. What is their first step? A. Bagging. B. Artificial hybridization. C. Insect introduction. D. Self-pollination. Correct answer is B. Artificial hybridization. Explanation: to develop disease resistance, the breeder must first introduce resistant traits into the crop's genetic pool through artificial hybridization. This involves crossing a susceptible plant with a disease-resistant variety. Bagging A is part of the process but not the starting point. Introducing insects C does not help with disease resistance. Self-pollination D maintains the status quo and doesn't introduce any new traits. Why are unisexual flowers favored in hybrid seed production? A. Prevents vine pollination. B. Enhances nectar production. C. Prevents self-pollination. D. Increases Cleistogamy. Correct answer is C. Prevents self-pollination. Explanation: unisexual flowers have either male or female organs, which naturally prevents self-pollination and promotes outcrossing. This is ideal in hybrid seed production where genetic mixing is desired. Wind pollination A is unrelated to flower sexuality. Nectar production B may influence pollinator attraction but doesn't affect selfing. Cleistogamy D actually promotes self-pollination, the opposite of what is desired in hybrid seed development. In what type of flower does autogamy occur despite being bisexual? A. Chasmogamous. B. Unisexual. C. Cleistogamous. D. JIPMER. answer is C. Cleistogamous. Explanation: Cleistogamous flowers are bisexual but never open, which enforces autogamy, self-pollination. Chasmogamous flowers, A, are also bisexual but open, allowing the possibility of cross-pollination. Unisexual flowers, B, have only one type of reproductive organ and cannot undergo autogamy. JIPMER, D, involves transfer of pollen from one flower to another flower on the same plant, not true autogamy.